Hello and welcome to another lecture. This one will be on the general parts of a membrane. This is Mr. Watkins. You can download or get a copy of this from the website. So let's get started here. The first thing we want to talk about is this little ball structure that has these two little leg pieces, these two little tail pieces sticking on it. This is called a phospholipid. Now, if you notice, there's lots of these all in the membrane. We've got basically a layer that runs this way across the top and a layer that runs this way across the bottom. Let's look at what the phospholipid, let's get a little bigger picture of the phospholipid so we can kind of see what it looks like. Now, a phospholipid is exactly as it sounds. It is made up of a phosphate group and a lipid group. Remember that a lipid has glycerol. Glycerol, 3-carbon backbone, and then 3 fatty acids. So here's the start of my glycerol molecule. You have your hydroxyls on one side, and you have some hydrogens on the other. Got to make 4 bonds. And what ends up happening is, you have your 3 fatty acids that come off here. And I'm just going to draw those fatty acids as a line so that to kind of save some time here. And in a phospholipid, you'll have 2 fatty acids that come off, and then where that third one would be, you actually would have a phosphate group that attaches to this third one. This phosphate group forms the ball shape that you saw earlier, so that's where we get the ball shape, and then these fatty acid pieces here that come off are the tail shape. So we really just kind of draw it something like that, where the ball shape again is the phosphate, and the bottom part here is the lipid, or more specifically the fatty acid. Now it's important to note that the phosphate group is polar, meaning it has the same type of characteristics that we had in water, where we have a molecule or a part of it that has a different charge than the other part. Phosphate being polar, water being polar. Remember, polar molecules like to dissolve polar molecules, so the phosphate group is going to be hydrophilic. In other words, it's going to like water because it's polar as water is polar. The lipid or the fatty acid is nonpolar, and so nonpolar likes nonpolar, but it does not like polar molecules. And so what will happen is this fatty acid doesn't like water, and so it is hydrophobic. Let's look at the model that we had of the membrane and go back and finish going through the rest of the pieces of that particular model. So here's our phosphate groups. If you'll notice, we have them all here. They're pointing on the outside or on the top, and then we have it all along here pointing across the bottom. I'm going to go ahead and label the outside and the inside part of a cell. This part here, this side, is going to be the outside part, and this is going to be the inside part. Inside perhaps maybe the cell. Outside and inside the cell, the cell is in a solution, and so that solution tends to be a polar solution or has water in it. The inside of the cell is made up of cytoplasm, and the cytoplasm is mostly water. So notice that the phosphates, which are also polar, are all pointing towards those sections that have water, and that the lipid portions of the cell membrane are pointing towards each other because they like each other. Nonpolar likes nonpolar, and they're basically making a boundary, almost a wall if you will, that puts a division between what's outside and what's inside. Remember that the cell membrane controls the movement of materials into and out of the cell, and this is one of the key ways that they help keep things from coming in or keep things from leaving the cell. Let's talk about the proteins that are on this particular diagram. We have a protein here. This is the big chunky piece. This protein is what we call an integral protein. And an integral protein means that it touches that nonpolar lipid layer. So this membrane here, membrane protein here, is also an integral protein. And this membrane protein here is also an integral protein. All because they touch the nonpolar lipid part inside that membrane. Also, you should note that because this protein is touching this part, the part of the protein that touches this inner membrane, this inner side, this nonpolar side, also must be nonpolar. Now these membranes, because they go all the way through from one side to the other of this membrane, are also called transmembrane proteins. Okay, so they're integral, just like this one is integral, but the thing is, because this one doesn't go all the way through, and these do, these two would be called transmembrane proteins. We have another type of protein here, another way of describing these membrane proteins, and that's this one here. This one's called a peripheral protein. And a peripheral protein does not go all the way through the membrane, doesn't go even into the lipid part. It's on the outside. And we'll discuss more about how they function as we get into photosynthesis and cellular respiration later on this semester. Another thing we need to look at is this piece here and how it's attached to this protein that's right there. So here's another one of these. Okay, and this is a sugar. These things right here are a sugar, probably glucose. We could say they were glucose, and they're attached to a protein. Now, most of the time when you see this, these proteins are going to be integral. They're going to be integral, and they may quite possibly be transmembrane. In other words, here's on the outside, here's that sugar attached to this protein that goes all the way through to the inside. So we have a sugar attached to a protein. Our prefix for sugar is glyco, and because it's a protein, we call them glycoproteins. One word, the sugar attached to a protein. These things act like receptor proteins. Now, receptor proteins are very important and they're very specific in their function. What they do is, either inside or outside the cell, they have these pieces that can attach to very specific molecules. They receive it. They pick those signals up. And that molecule combining and attaching can cause a change in the protein that's attached to it, or send that signal through the protein to the inside where maybe there's a peripheral protein that picks up that signal and causes the cell to perform some function. Maybe that's to release a chemical outside the cell. It could mean that it could make a new protein, but there's a lot of different functions. We also have a sugar, same kind of thing that's here, a sugar attached here, but this one's attached to the lipid part, and since it's still a sugar attached to a lipid, we don't call it a glycoprotein, we call it a glycolipid, and it has the same function as a receptor protein. Now, our final piece that we're going to talk about in here that's in this membrane is this piece right here, these little pieces right here, and this is cholesterol. Now, cholesterol typically, in a biological sense, will increase, in the membrane, will increase rigidity. Now, it depends upon the temperature, but for what you need to know at the moment is that it's going to increase the rigidity. As the temperatures drop, it increases the rigidity of this membrane. Still allows it to be fluid, but it gives it some strength that may not be there at different temperatures, and that's the function of cholesterol. Now, we'll talk more about those other pieces. There is two other types of transmembrane, or actually they can be integral proteins, and they are transport proteins, and there's two basic types you must be aware of, and that is a carrier and a channel protein. You need to watch the lecture on membrane transport, or transport across the membrane, and that's where I'll go over those two different types of membrane proteins, the carrier and the channel protein. Make sure that you can label a drawing very similar to this, label all the general parts that make up a membrane. That's it.